Thank you for that introduction, Gaute. What I want to show you is our work on how current source densities can reveal deficiencies in a model, and also how analyzing current source densities can aid model development and provide more constraints in your optimization. Traditionally, models of neural networks have focused on reproducing spiking activity and put less emphasis on reproducing extracellular potentials. And that has especially been the case if it's a larger scale network where the complexity and level of detail makes the calculation of extracellular potentials very computationally expensive. But only using spiking activity to optimize and validate a model can have important limitations. And for one, even the most state-of-the-art electrophysiological recording devices can only record from a relatively small number of cells in any given area. And it's only a tiny fraction of the total number of cells in that area. So even if you assume that there are no systematic biases in your recording, the sample can often be non-representative. And there may, of course, also be biases in the recording, such as strongly firing cells being picked up more easily and so on. And the local field potential, on the other hand, measures the activity of entire populations of cells. And as such, it can give complementary information about the recorded neural activity that is not affected by the same sampling problems that you have when only measuring spikes. Now, the model that we've used in our work is the biophysically detailed model developed at the Allen Institute. It's a model of the mouse V1. And this model was made publicly available in 2019 and presented in Neuron in 2020. It consists of 230,000 neurons, of which 50,000 are biophysically detailed. And those 50,000 constitute a core surrounded by leaky interpret and fire neurons. The experimental data that we are using is the also publicly available NeuroPixel dataset released by the Allen Institute. It has recordings from 58 mice that are exposed to a number of visual stimuli. And in each mouse, 6 probes are recording spikes and LFP from the various visual areas. In our work, we have been focusing on the response to full field flash stimulus, even though there are many other stimuli you can analyze data from. And when we compare the model to the experimental data, we don't use the LFP directly. We use the current source densities instead, because it's easier to interpret and it's derived from the LFP. And the relationship between the LFP and the CST is shown here. The CST is the double spatial derivative of the local field potential multiplied with the electrical conductivity. And the CST is assumed to be constant in the plane, so you only need to take the spatial derivative in the depth direction denoted set here. And to further illustrate the relationship between the LFP and the CST, I plotted the LFP from an example animal in the data set at the top left here, and the LFP from an example simulation at the top right, and the respective CST from that LFP at the bottom left and bottom right. And the way to read these CST plots is that the blue blobs here constitute current sinks. So if the current sink stems from input from excitatory synapses, this sink shows where positive ions enter the cells. While the red blobs are current sources, and the interpretation of them depends on whether the synapse is excitatory or inhibitory, but if it's an excitatory synapse, it shows where positive ions leave the cells. and enter the extracellular medium. Now I'll go more into detail about what information you can get from the CST plots. And what's shown in these panels are the trial average CST in response to a full field flash for different animals in the LN dataset. And the depth axis goes along the depth of V1 with the different layers of V1 annotated in the leftmost plot here. Now, as you can see, there is substantial variability across animals in these CST patterns, but there are also some prominent features in common. In particular, there's a layer 4 sync starting around 40 milliseconds after the onset of the flash, followed by a layer 4 source, and it's more sustained and with a sync above and below it in layer 2-3 and in layer 5/layer 6. And this pattern is found in almost all animals, although there are some exceptions, such as this rightmost animal here, where you do have the layer 4 sync, but you don't have those sinks and sources following the layer 4 sync. Now we want to identify the common features across animals in a more objective way. And to do that, we stack the trial average CST from all animals together and do PCA across them. And the first principal component, which would then constitute a weighted average of the CST from all the animals, explains about 50% of the variance. And we see that it has the same pattern that we observed in the individual animals. And so the CST of this first principal component would then constitute the target that we want to optimize our model towards. Now we need a metric to quantify the variability in the CST patterns across animals, and also to quantify how close the model is to this target CST of the first principal component later on. And for that, we'll use the Wasserstein distance. The Wasserstein distance tells us how much we have to move the sinks and sources of an individual animal to get them to match the pattern in the CST of the first principal component. And the more you have to move the sinks and sources, the bigger the Wasserstein distance you get. And thus the more different the animal is from the PC1 pattern. So when we plot the obtained Wasserstein distance between each animal and the PC1 pattern, we get to a distribution of Wasserstein distances in the experiments. The stars in these box plots, in this box plot, denote the example animals at the top. And we see, as expected, based on the qualitative comparison, that the rightmost animal is indeed an outlier. It's the purple star at the top here, while the other animals that have CST patterns that resemble the CST pattern of the first principal component have smaller Wasserstein distances. And now that we have a target and a metric in place, we can start to see how well our model is doing. The CST from a simulation is in the middle plot here, where we used experimentally recorded spike trains from LGN as input to the model. And the configuration of the network was changed from the state it was in when it was published, when it was made publicly available. But still, the resulting CST does not resemble the experimental CST at all. There is a sink in the deeper layers, but the sinks in layer 4 and layer 2-3 are the source following the layer 4 sink are either completely absent or way too weak relative to the deep layer dipole to be visible. And when we compute the Wasserstein distance between this simulation and the pattern of the first principal component, and compare it to the distribution of Wasserstein distances for the animals, we see that the model is among the outliers on the animals. Now, as mentioned, we wanted to reproduce both spikes and LFP. And when comparing the spikes of the model to the spikes of the experiments, we look at the laminar population firing rates. In the panels here are the firing rates of the excitatory populations in each layer, and the inhibitory populations of all layers are together in the rightmost plot. And the black line is the average firing rate of cells from all animals together. And to quantify the experimental variability, we use the Kolmogorov-Smirnov similarity, or KS similarity for short, and the correlation. So what we compare here is the population firing rate of each individual mouse to the average firing rate over cells from all mice. So the average, or the black line here, in each population is again the target, although it's not the weighted average as it was for the CST. And this way, we again get a distribution that we can use as a reference to see if our model is within the experimental variability. That is, if the population firing rates of the model are indistinguishable from the population firing rates of the mouse in this data set. The KS similarity here gives us a measure of the similarity in magnitudes in certain time periods, such as during the baseline or at the peak, while the correlation gives us a measure of the similarity in the temporal profile. So when the peak occurs in this 100 millisecond time window, for example. And when we compute the population firing rates for the simulation that I showed earlier and compare them to the experiments, that's a blue line in these firing rate plots and the blue diamonds in the metric plots. You see that the model is actually already not doing too bad. With the exception of the layer 6 inhibitory cells and the inhibitory populations, where the firing rates are excessively high, the model is mostly already within the experimental variability. And with a few adjustments to the synaptic weights, that's shown in green here, the resulting firing rate after adjusting the synaptic weights. Specifically, what we did was that we reduced the weights from excitatory populations to the inhibitory populations to lower the inhibitory firing rate, and then reduce the synaptic weights between excitatory populations to compensate for the lowered inhibition. And that was enough to get the model within the experimental variability in all populations on the different metrics. So now it appears that we have sort of already reached our target with respect to the firing rates. And the question is then, what does the CST look like? The answer is not very good and not really all that different from what it looked like before. The upper layer sinks, so the CST from the simulation is shown in the middle, and the upper layer sinks in layer 4 and layer 2, 3, and the upper layer source is still absent or way too weak relative to the deep layer dipole. And the Wasserstein distance between the model and the pattern in the first principal component is still large. So this is already an interesting observation. With the adjustments we made to the synaptic weights, the population firing rates of the model can't be distinguished from the population firing rates recorded in an experiment, while the CST is almost unchanged and not close to the experiments at all. And this shows that the CST, or the LFP, and spikes really do represent different information in their recordings, and thus that you learn something more by looking at the CST. And the question now becomes, what determines the observed CST pattern, and what adjustments can we make to get a better correspondence between the model CST and the experimental CST. So one thing we can do is to try to move around on the positioning of the synapses. So one advantage with the model is that we can actually disentangle the different contributions to the total CST, the contributions from the different populations to the total CST. So in these plots, I show the contributions from the layer 4 inhibitory cells. And when I put, for example, all synapses onto only the apical dendrites, all excitatory synapses only on the apical dendrites of the layer 4 excitatory cells, or all excitatory synapses on the basal dendrites of this population. And we see that when all the synapses are on the apical dendrites, the sink is at the top, or in the middle rather, while there's a source above and below it. While if all the synapses are on the basal dendrites, there is only a sink at the bottom with the source above it. Now, the firing rates will be affected by modifications that are this drastic, but you can either way just compensate for that by adjusting the synaptic weights if that's needed. We can, of course, not do anything we want with the synaptic placement. We are constrained by the anatomical data and what is biologically reasonable. So we have explored many different possible combinations of synaptic positioning within these constraints, but neither of them were sufficient to reproduce the CST pattern of the experiment. So something else was missing. And that something else turned out to be the feedback from higher visual areas. We have recordings from the higher visual areas in the mouse visual cortex, and we plotted the firing rates of these areas together with the firing rates of LGN and V1. The firing rate of LGN is shown in blue, and the firing rate in V1 is the orange line. And when we look at the firing rates in, if you follow the red line and the green line and the purple line, the firing rates in different higher visual areas, we see that they generally peak around 60 milliseconds. And that's the first peak as well. And when we look at the CST pattern in the experiments again, we notice that there are strong and sustained sinks and sources that pop up around exactly 60 milliseconds. And one more thing that is important to mention is that there's also a strong source, a sink in the layer 1, layer 2, 3 region, an area that is known to be a region where input from LM, one of the higher visual areas, are focused. So up until this point, the model only had input from LGN and background noise representing input from the rest of the brain. And we decided to try to feed the recorded spike trains from LM, one of the higher visual areas, into V1, which would then represent the feedback to V1. And when we did that, we saw almost an immediate improvement in the correspondence between the model and the experiments. The CST of the model with the feedback added is in the middle. And we see that we now have a strong sustained source in layer 4 with a sink above in layer 2, 3, and a sink in layer 5/layer 6 as before. And also a layer 4 sink, although that is a little bit weak still. And when we compute the Wasserstein distance between the model with the feedback added, we see that the distance now is almost as small as it was for some of the example animals shown at the beginning. And when we compute the population firing rates after adding the feedback, we see that we are still within the experimental variability on the different metrics. So We can now say that we have a model that can reproduce both spikes and LFP as they are observed in the experiments at the same time. And that's another interesting observation, because the firing rates were, after all, already within the experimental variability before we added the feedback. So it was not apparent by looking at the firing rates alone that the feedback from higher visual areas was missing in the model. That only became apparent when looking at the CST. And this is something that further supports the view that spikes and LFP really represent different and complementary aspects of neural activity, and that by computing the LFP and the CST, you can reveal deficiencies in the model that you wouldn't necessarily see that easily by only looking at the spikes. And as mentioned, one of the advantages of the model is that you can disentangle the contributions of different populations to the CST and the total CST. So in the figure to the left here, you can see the contributions from the excitatory populations in each layer and the inhibitory populations in each layer to the total CST. Some of these contributions will cancel out when we sum them to the total CST, but we can, for example, see that it's the excitatory layer 6 population that is responsible for the deep layer dipole observed in the total CST plot to the right. Now, we've used this to determine what adjustments to make in this project. And we can also use this approach to guide our future work on this model. We can look at the distribution of these sinks and sources and try to determine how they might sum up or cancel out to produce the specific patterns in the total CST. So to summarize, the original model did not reproduce neither the CST nor the population firing rates in the experiments. But by adjusting the recurrent synaptic weights of the model, the population firing rates match the experiments. But the molten CST was still largely unchanged and did not reproduce the experimental CST. And the CST of the experiments was only reproduced after introducing the feedback. And together, these findings show that the LFP and the spikes really do represent complementary aspects of the neural activity. And if CST is used in addition to the spikes when optimizing and validating models, it should put stronger constraints on model configuration and on parameters. So lastly, I just want to thank Anton and Gaute and Christoph for all advice and guidance on this project. And I also want to thank Espen Hagen and Alexander Stasek, Kyle Dye, Josh Siegel, and Yasan Bile, as well as all members at Simpla here in Oslo for help and explanations and other important contributions to this work. Thank you for your attention.